2026 Spring Project Talk Series. I'm Deanne Krupa, the Grant Manager with Innovate Bio at Austin Community College in Austin, Texas. It's a pleasure to facilitate this presentation. Both Innovate Bio and this series of talks are funded by the National Science Foundation. Before we begin, I have a few housekeeping announcements. The two presentations will be back-to-back, followed by a Q&A. Please post questions for our speakers in the chat, and also additional information on these two ATE project talks, as well as future project talk events, can be found on the Innovate Bio calendar of events, which we'll put that in the chat. We respectfully ask you to complete a survey at the end of the presentation, which will help us in planning future events. And also at the conclusion of this year's Spring Project Talks, participants will receive a certificate of attendance. At this time, please join me in welcoming our first Project Talk speaker, Sandra Porter. Her ATE project hosts a national anti-engineering hackathon that brings students, faculty, and industry mentors together to participate in developing and implementing research projects in antibody bioengineering. Sandy, take it away.
Thanks, Deanne. I'm going to share my screen here. There we go. Okay, well, our official name, Developing Cures for Antibody Engineering, cures being course-based undergraduate research experiences, though you'll learn it goes way beyond that. The people who have been contributing the most to the project are Maggie Bryans from Montgomery County Community College, Todd Smith from Digital World Biology, Sheila Vemu from Waubonsie College, Erin Kamajaya from Los Angeles Pierce College, and Alyssa Dillman, our hackathon expert who's been telling us all about what to do and how to do it. And there's lots more people, as you're going to learn. So today, I'm going to give an overview of our hackathon work over the past 5 years, what we've learned, and its application to the ATE community. First, everybody asks, what is a hackathon? A quick, short definition is that it is an event where teams work together for a short time to create products. Okay, so we're going to be making something. It's also a kind of professional research activity, very interactive, intense. People work in teams and present results. You really learn, you can learn a lot by participating in a hackathon. You can work with a very diverse group of people from all over the country, build on previous work. As the NISCHO team has done, you can build community. And unlike a conventional workshop, team members have assigned roles. There's a team leader. There can be writers, data hunters, coders, slide makers, presenters. And this isn't a comprehensive list of all the kinds of things that people might do and people might be involved in. By the way, before I forget, there is a lot of information on each slide. These recordings of these presentations will be made available on the Innovate Bio website, and I'm happy to share the slides with anybody as well. Okay, there can be some misconceptions about hackathons too. They do not require coding. Hackathons got started in the software engineering community, but they can be applied with all kinds of things, wedding planning, whatever. A broad group of activities. They don't require coding. They don't have to be a competition. They can be, but they don't have to be. You don't have to be an expert to participate in a hackathon. They don't have to have lab work, and you don't even have to travel because they can be virtual. What they do require, though, is they do require time, and they require a lot of communication and participation. If you don't attend the meetings on the first day and you get lost, it's really hard to get back into it and feel like you can do anything. They require a lot of preparation and teamwork and organization. So we're going to take just 1 minute and listen to something we created in last summer's hackathon. And now, there we go. So what you just heard was a nanobody sequence. Nanobodies are short single-chain antibodies. This one is from humans, and you heard violins at the spots where there are two alanines. You heard the his tag, that was the drum. This was from a team that decided they wanted to sonify antibodies, which is kind of a different thing. And I need to move this. Okay, there we go. Just to illustrate some of the principles about hackathons, people had different roles. Thomas Honorato from LaGuardia Community College was the team leader. We had coders, we had music liaison, we had database explorers, we had somebody design the logo, somebody working on curating the nanobodies and designing the slides. And this team actually did use a lot of different kinds of software. So this is a project that was made by the Nature's Bards team. Now, you wouldn't necessarily think about converting DNA sequences to music. It was kind of cool. But they thought this could be a way to better understand differences in sequences between antibodies and possibly use that in antibody design. Okay. So what other kinds of products have we been making through our hackathons? We have also been making research projects, protocols, data sets, games, other kinds of materials. Mostly people have been working on developing the supporting materials they need for course-based research projects and for independent student research projects. And these are all students who have participated either in the hackathons or they've worked on the research projects that were developed in their classes afterwards. Okay, we've also had a lot of publications. 13, really, 9 were in peer-reviewed journals, 3 were published in Cubes, and 5 of these had student first authors. So it's been a productive thing, and you can look at all of these at our website, antibodyengineers.org. So who has participated in our hackathons? Last year, we're really 8 to really kind of 10 teams, and the team leaders were from 8 different states. We even had one team from Hawaii, which made it interesting with time zones. Last year, there were 62 people, and a third of them were people that were returning for a second, third, fourth, or even fifth time. And they came from 16 different states. So you can see here about slightly over half of our participants have been students. A large number have been faculty, and then we've had a good number of scientists from either industry or academia. The majority or largest group of people participating have been from community colleges, but we've also had people from four-year colleges, high school students and faculty, universities, and, as I mentioned before, industry. And when I say industry, I'm really defining industry, government institutions, research institutions, any kind of scientific enterprise. So these are a lot of work. People do need to participate full-time for the amount of the hackathon, 3 to 4 days. Why do they do that? Well, it turns out that our faculty participants have really wanted to do this because this presents an opportunity to workshop ideas for their research projects. They don't always get to work with other faculty or students to have students test things out. So this gives them a chance to try things, develop the materials they're going to need, see how it works, see how they can communicate their ideas and if it makes sense. And they learn a lot of new techniques. Our industry and academic scientists have said that they participate in the hackathons because it gives them a concentrated block of time away from all their other responsibilities to focus on learning new techniques. And they like to work with others. Now, the students, many of them are participating because somebody told them to. So it can be a little bit different for them, but they do see it as a learning opportunity. And many do get excited about the possibility that there might be publications, there might be research projects coming after it. So, and they don't always know what they're getting into. So they participate for a variety of reasons. All right, our teams that we had last year, we had Maggie Bryans and Lori Kelman were leading teams working on NIST CHO cells. They were developing all kinds of protocols for all kinds of experiments to test protocols and media for this special cell line that was developed by NIST for producing antibodies. Todd and Eric Olson from Borealis were working on different AI models to make antibody predictions and validate them. Erica Lannon for antibodies in cancer. Danny Kaner's team was working on developing worms that would digest styrofoam. Thomas Ornato, you heard a little bit about their project. Melanie Stegman was working on a game for escaping the thymus. Dylan Bolseco was using AI as a research team. And then Christy Martinez, she's a high school teacher, was developing a team to look at how to better communicate immunology concepts about antibodies to high school students. So we had a wide variety of teams doing a lot of really fun things. Okay, so this was our 5th hackathon, and during the time we've been doing this, we've changed a lot. I kind of highlighted some of the things that changed the most. We tried to do a lot of things up front with the announcements, with who we accept and how we can make sure we confirm that people are going to be attending, how we assign people to teams, finalizing the schedule. We set up a whole Slack instance. Everybody has Slack accounts, and we have a pre-hackathon orientation to let people know that, again, this will be a lot of work. So some of the things we've really changed, I mean, that was a thing we added. We have added team leader meetings because we find that although faculty, they know what it's like to manage a classroom, they do not have a lot of experience with managing people from a wide, diverse group of backgrounds. And it can be a little bit challenging. So we talk about some of those challenges, setting expectations for what the presentations will be like, answering questions, what to do if somebody doesn't show up, things like that. You notice we have a lot of presentations. We try to give everybody who's participating a chance to present. Here in our schedule, our first day, it's kind of logistics and everybody gets going and starts working. The second day, though, the teams present the work they're going to do and their plan. The third day, the teams present any kinds of problems that they're having, and everybody else can help with solutions. The last day is largely devoted to final presentations where the teams present the work that they created and what they've done and what they might do next. Over time, we used to have a lot more guest speakers originally, but then we learned that no, people really do want to have the time to work. And if we give them too many speakers, it gets really frustrating for them because they don't have the time to do the work and do their own presentations. So we had on Tuesday and Wednesday, we had 2 guest speakers each day, but it was only for about an hour that they talked. So it was a little bit less time. We also added a speed networking session this year, which was very successful, and a lot of people commented that they really appreciated doing it, especially our high school teacher, who noted that students don't often get a chance to just talk with industry scientists or talk with faculty from colleges about what they might want to do for applications to colleges, or what a career path might be like, and that this was a really unique and unusual opportunity that she thought it was really valuable. We also found one of our scientists who has told us he hates these kinds of things. He hates networking, so that he really enjoyed it too, and found it a good time. Karen Leung, who is here, is really an expert at speed networking. She was the one who set everything up and managed all the breakout rooms for us. And if you're thinking about anything like this, really talk to Karen, because she's great. Okay, I also want to point out that one of the roles here that we have a special meeting for on Tuesday is a writers' meeting. So it is really important that we document our work so that we can come back to it later and we can benefit from it. And so one of the most important team roles that every team has to have is to have someone as a writer. And so there's a special meeting for writers to talk about that and understand what they're being asked to do. Okay, so what did we learn a little bit more after 5 hackathons? People are surprised by the workload, especially the students who don't believe it. Always tried it, but we get a few who are trying to work full time during this and doesn't work, or they're trying to take classes during this, doesn't work. So that can be an unpleasant surprise for them. But we found for the faculty and the scientists and the students who really participate, the work is the attraction. Being able to work with other people from all over the country with a variety of skills and expertise and learn about what other people are doing and present their work and hear other people present their work. They really like that. We also learned some good tips for team leaders that they can do really well when they recruit team members. One team with Maggie Bryans and Lori Kelman, they have had practically the same team for the past 3 years working on the NISCHO cells. So they've come to work really well together. So they can find people that do things. We also find people are developing skills in time management, communication, time zone math. It's really tricky sometimes to make sure everybody has time for lunch when you're working with people on different coasts. And we're finding students are posting testimonials on LinkedIn, which is kind of a cool thing as well. Okay, lastly, we do not yet know if we're doing a hackathon this summer, Antibody Engineering Hackathon. We hope to find that out soon. But whether or not we do that, there is going to be an Innovate Bio Hackathon. We're calling it the Innovate Bio Big Ideas Hackathon. This will be 3 days, because we know it's hard to take 3 days off to do anything. It will be virtual, but it will give you a chance to work with your colleagues on developing ideas about biotech education. This will be Monday, June 29th through Wednesday, July 1st. And some of the potential projects will be people working on ideas for the next NSF ATE Biotech Center. The next grant for the center that happens after Innovate Bio is due in, I guess, a little over 30 months. So whoever wants to be writing that grant, they should be starting to work on it now. And it would be great if they wanted to come participate in the hackathon. We will have teams working on ideas for student recruitment, the value of 2-year degrees, AI and teaching, biomanufacturing, automation, high school partnerships, and other ideas as well. And you can find out more information about that at innovatebio.org. And that is it. And I'll answer questions when it's time. Thanks.
Thank you, Sandy. Looking forward to the Innovate Bio Hackathon coming up. And again, links will be posted in the chat. Our second Project Talk speaker is Katharine Marsh, and joining her is Gerson Baggy. Their presentation will discuss their efforts with student recruitment into their biotech program, highlighting one of their most successful strategies of leveraging high school partnerships to recruit dual enrollment students. Welcome, Katharine and Gerson.
Hi, thank you so much. I'm Katharine Marsh. I'm at Compton College. I'm the PI on this grant, and joining me today is my dean, Dean of STEM. Um, I want to first talk about the rationale behind this grant. My slides are not advancing. Oh no, why is it not advancing? Okay, sorry. Try again. I want to first talk about the motivating rationale behind even starting our biotech program and writing this grant. We initially did an analysis of the job openings in this area through um, the Center for Excellence, and we found that the number of jobs greatly exceeded the number of qualified applicants based on how many educational awards were being conferred. So it's nearly a 1,000 position gap in specific to our local area. On the right, I also included a map. This is of all the local biotech companies in our area. You can see Compton. The word is blocked a little bit by one of the blue dots indicating a biotech company, but there's a lot of companies in the area. And we also looked at what percentage of our students are receiving financial aid. And we found that 70% of our full-time, first-time degree-seeking undergraduate students receive need-based Pell Grants. And we also found data that 30% of children under age 18 in the city of Compton are living below the poverty line. This is twice the level of the LA County in general. So that's the population of students that we're serving. I looked for the data about what kind of salary potential do students have when they complete either a biotechnology or a biomedical technology certificate at a California community college. And the data shows, so I set the graduation at 0.0. 2 years prior to graduating, the average salary was $20,245. And on the timeline 2 years later, with the certificate, their average salary was $46,499. And then 5 years later, it was $58,624. So it seems like there is really potential for our students to really move up the socioeconomic ladder. So this was kind of our motivating rationale. So our initial ATE grant was a new to ATE to get our program started. And we built out elements that we thought would strengthen our program. We really wanted to have strong connection with industry. So we built an industry advisory board. We were bringing in guest speakers from the biotech industry, and we do tours to biotech companies, to our local biotech companies. We thought it would be important to give students options for how much education and training they wanted to pursue. So we designed our program to have 2 stackable certificates. Students can take 2 classes and get a lab assistant certificate, or they can keep going. They can take all 5 of our biotech courses and get a biomanufacturing certificate. Or they can keep going, take their general education, take chemistry, statistics, and get their Associate of Science degree in biomanufacturing. Oops, sorry. We wanted to facilitate students being able to buy course materials without money being a barrier. So we use all open education resources in our courses. So the textbook, the lab manual are free and available online. If students want a print option, they have a low-cost print option as well. We really wanted to make our program relevant, so we focused on real-world applications. In our courses, students are doing CRISPR, things they've heard in the news, forensics, and we encourage students to come up with their own projects, what they want to work on, so they can pursue what they're interested in. We thought it was important to support our students with a biotech peer mentor to talk to them and see where they're at and what challenges they're facing. And then the biotech peer mentor comes back to me and whatever the students need more support with, whether it's resume writing or if they're facing some imposter syndrome or things like that, then we bring in resources from our campus to support our students. We have a course on quality in a regulatory environment, and we really wanted to bring in a relevance. So using our grant funds, we hired a case study writer, Jim Hewlett, who's an expert in biomanufacturing and also writing case studies. He looked at our course outline and he analyzed what are our learning objectives and our student learning outcomes. And he built a series of case studies around that, and they were really well scaffolded with over a few day period, giving students a little bit more information, and it was really a positive experience for our students. We also want to introduce students to research early on. So in our Biotech 101 course, students do a CURE, which we just heard about, the course-based undergraduate research experience. We did one on DNA barcoding, and the advantage of DNA barcoding is that all students in the class follow the same methodology, but they can pursue different projects within this. So everybody's doing the PCR and the gel, but they can ask different research questions or study different types of species. They can look at fish and investigate seafood fraud, or they can do a project looking at identifying species of plants using DNA sequencing. And they can do if they have a question about invasive species, they can design their project around that. So I really like this specific cure because the setup is more simplified since everyone's doing the same protocols. Maybe their primers are different, but they can ask their own questions and design their own research projects. And then, of course, this is a CTE, a career technical education program. So the goal is to get students into jobs. So we built a strong connection with industry, and we bring speakers in to talk about jobs and how they got into their career. And we have collaborations with companies that are willing to offer our students internships. So that was our new ATE grant, new to ATE grant. And what I want to focus on is some of our strategies for student recruitment. That's been a challenge for us, and talking to my colleagues, that's a challenge for other biotech programs as well. It seems as though the outcomes are generally really positive across the board in terms of getting students into jobs that pay a living wage. But building awareness and recruiting students has been a challenge. So some of my strategies were to do classroom presentations to students to talk about what we're doing and what our courses are and what the job looks like. We also have been emailing students who apply to our nursing program at our college. So the nursing has an application process, but we have many more students who apply than actually are admitted and matriculate. So there's a pool of students who are interested in doing some kind of vocational training, and so and maybe they didn't get accepted into the nursing program right away. So we're trying to capture some of those students, and we have been able to get students from that pool. Another way that we're exposing students to what biotech is through summer biotech boot camps. We've done these both at our high school site, and we've done it at on our campus, and it's been a positive experience and rewarding experience for the students who did that. We really worked with the high schools to figure out what's the best way to do this. So in terms of scheduling, like when during the summer, we really worked with them, and the high school was able to get their foundation to pay participants a stipend for doing it. So that helped with our enrollment, and it helped for students who need to work or things. Having the stipend was a benefit. And they were required to do some online work on Lab Exchange so that they can learn some of the background and the conceptual pieces of the experiments. And so they had to attend every day, do the hands-on lab. We did a company tour within that week, and then they also had online work to do. And so if they fulfilled all of those things, they earned a stipend at the end, and it was paid out of the high school foundation. I've also attended college and career fairs geared towards high school students. And I've met a lot of high school teachers and talking to them about what their perspective is. I had a high school teacher at an alternative high school tell me that he would rather move his students to an online high school and get them into these vocational training programs at community college. He worked historically, he's worked more with like welding and HVAC, but he said it's more important to get this training. These students are ready to work, so he will even move them to online school so that they are able to attend Compton College and get that vocational training. I also have presented to our counselors at our college because by talking to the counselors, they interact with a lot more students. I can do presentations and try to reach students at tabling events, but they really have more dedicated time with students and they interact with a lot of people. So, and I've told counselors at our college, like, please refer students to me. If they're interested, I'll meet with them and talk to them. And that has panned out. I have had counselors do that. And then what I want to focus on is one of our successful strategies, which was recruiting high school students. So we did this a couple years ago. Let me tell you some of the details about how we set this up. So I collaborated with the career pathways coordinator at our college, and the high school had a CTE coordinator who was really interested in this. So we both went to the high school. I did presentations to different classrooms to talk to them about this. And their students already had some exposure to biotech through what's called Project Lead the Way. So that's a CTE focused curriculum. So the high schools were already doing a little bit of this. So it wasn't totally foreign to them. So I think we had an advantage in that regard. But we helped students. We went there. I presented. I talked to the teachers. We polled students how many are interested. So it was helpful to have that number to bring that back to our college to justify. Look, we've got enough students. Let's offer this class. The coordinator that I was with was able to help really get students starting with the paperwork, the enrollment paperwork. So that was helpful to not just present, but right there and then get started on that and start enrolling them. So we worked with the coordinator to figure out scheduling. We decided to offer this course on our campus, but we offered it as a hybrid course. So the lecture was fully online and asynchronous. There were no scheduled meeting times. We picked the lab to take place in the afternoon. They had a minimum day, so they had early dismissal every week. Every Wednesday, they got out of school at 1:30 instead of 2:30. The high school coordinator, so then the benefits students, they get waived tuition because of the agreement that we have for the dual enrollment students. The high school coordinator paid for a bus to pick the students up at their high school and bus them to the campus, to our campus. So the bus stopped at 2 different high schools, picked up students, brought them to our campus. They were there for 3 hours to do the lab every week, and then the bus took them back to their high schools. We also worked out to get the free printed lab manuals. We're using the LibreText lab manual that the Orange County Community Colleges developed. And as a result, we completely filled this class. It was like kind of the tail end of COVID at the time, so the caps were the cap was 23, I think, but we totally filled the class. It was almost all high school students who had heard my presentation, who had someone there right next to them to help them start filling out the paperwork to enroll, to learn about the benefits, to have this coordinator figure out the bus and figure out the scheduling. It worked, so we filled our class. And I want to share with you one of the outcomes. A couple years later, a student emailed me. My slides aren't advancing again. And I want to share her quote. Sorry, let me try this again. Sorry. Oh my gosh, why are they not advancing? Oh, let me try using the Zoom controls instead of the PowerPoint ones.
I'd help you, but I don't know how to do it.
Thank you, Lydia. A student emailed me a couple years later. She wrote to me, she said, I would like to express my gratitude for taking the time to write me a letter of recommendation for my college applications. I got into all the schools I applied to. I'm currently a first year at California State University, Long Beach, majoring in pre-biomedical engineering. Your course was a highlight of my high school career that led me to pursue my major. Unfortunately, I could not enroll in your spring courses as it conflicted with my class schedule. However, I would like to continue taking your courses. Will you be offering courses during the summer or have any internship opportunities available? And I did invite her to come in and speak, but she's taking an exam right now, so she wasn't able to. But she said it was, it's surreal. She said where she's at and the experiences, it was really a turning point for her. But this allowed us to identify a problem. We did the first class for high school students in the afternoon, but the next class has more hours and more lab, and it didn't fit well within the high school schedule. So with this grant proposal, we looked at what can we do to figure out how we can build out our dual enrollment. So I really wanted to listen to what are the high schools interested in, what's going to benefit them, what do they need? And I found out that they're interested in their metrics. So there's a California school dashboard that the schools get like a color-coded and number associated with different parameters. So he told me he's specifically interested in the college/career indicator. So in my proposal, I researched this and I looked at what are the benchmarks, and I built out how to meet those. So some of the things are like just being a dual enrollment course. Does it transfer to the Cal State and the UC? And our Biotech 101 does. And it fulfills the general education of life science with the lab. There was a capstone component, so we looked at how we can build a pathway of courses with a capstone. So we looked at that and how we can meet their metrics, just like community colleges are. We have funding formulas, and so the numbers matter. So same thing for the high school. So for someone else looking to do dual enrollment, these are some considerations. Scheduling, so we did it at our Compton campus. We're looking at possibly offering it at the high schools. So that's one consideration at the college or at the high school, or the timing. So we did it during their minimum day. There's also options to do like 0 hour or to build it into the high school schedule. The modality, so we did leverage online learning to better fit into their schedule. Let's see how I do with that. Another thing is looking at who is going to teach these courses. So in California, to teach a college course, anyone teaching it has to meet the minimum qualifications. For biotech, it's a bachelor's degree in some kind of biology field and 2 years of work experience. And so if there's any ambiguity whether the person would be qualified, at my college, we form a committee and we determine if they meet the minimum qualifications. One caveat to consider, if the committee determines that they meet the qualifications, they can teach any of the courses. So I saw this with biology. My colleagues were saying maybe this person could teach non-majors but not majors, but that's not how it works. If you say that they're able to teach them, they can teach any of the courses. So keep that in mind for anyone doing this. I talked a bit about student recruitment, so I'll kind of gloss over that. We also looked at how we can have a pathway with articulated on-ramps and off-ramps, articulated with our universities, and also more in California, more community colleges are offering a bachelor's degree in biomanufacturing. So we have 2 in LA County that are just getting started with that. So we're looking at how can we make sure our courses are articulated to give students the options to keep going if they want to go to get their bachelor's degree. We want to really bring in the teachers and counselors at the high school to really let them know what biotech is. And one challenge, I think, especially if we were to do courses at the high school, would be the support for lab preparation, the work to aliquot the reagents and make the buffers. So that's something we're thinking about at this point. So one thing I thought about is if we time it so that our Biotech 101 is offered both at our college and at the high school. Our lab technician would just do one prep, maybe just more materials, but it would be still a little more streamlined. And then we do have an internship program. So we have an internal internship program. So our student workers could potentially, that could be the time and the labor to prepare the lab materials. Other considerations are equipment, not only the cost, but also just the space considerations of bringing in the equipment. What I've seen, our high school, sometimes it's a shared lab. It's not, they don't have like a dedicated biotech lab. They're also doing physiology and other things in the same space, so that can be tricky. And then facilities, they need to have enough bench space, enough outlets to plug in things, and then they need to have some, sometimes the high schools maybe don't have as much safety things, definitely a sink or eye wash or safety shower, things like that. So these are things to think about for people who would want to do this. And then finally, my advice for how do you equip a biotech lab on a limited budget with limited time. There are low-cost kits and the associated curriculum available. Probably a lot of people are familiar with Biotech. I saw Yingsu is here, the Bay Area Bioscience Education Community. They do a lot of work with high schools. Their kits are very low cost, and so that's a good option. Also, when we buy equipment, I have the field application specialist come during class time. So the students hear the talk that the field application specialist is giving. And I've really gotten a lot of buy-in from them too. I have one guy from Fisher when I bought a flow cytometer. He helped me build out like a protocol. I wanted to do some T-cell activation. He really helped me develop a protocol. He gave me a 4-color antibody panel for free. And then he comes back every time we offer that course and he gives the talk to the students and he's there supporting them through doing this protocol. We get primary T cells and we activate them. And he really worked with me to build out this protocol. And then he also let his boss know in case he moves around or whatever that there's some buy-in from them too. Like if someone else were in his position, they could keep sending somebody back. He doesn't charge us. He comes back every year to give this talk and to do this project with their students. Also, buying equipment, one consideration is to look for equipment that gives you unlimited licenses versus just one license, because a lot of it is, like for example, with chromatography, a lot of it is setting up the protocol and analyzing the data. Students can do that at home or as an assignment. So I think an important thing to look for is unlimited software licenses for equipment. Also, requesting product demonstrations at the time from sales representatives. So could be a way that they can come and demonstrate the equipment. And then when there is a budget, you'll be ready to go with what you want to buy. But students can get that experience and see the equipment through a product demo. Yingsu did a lot of work with the Bioscope supply chain. So having, like for example, our student workers at our college build out some of the materials and then somehow invoice the high schools for that or something could be a possibility. And then there's a lot of companies that have offered us donations of equipment. The Amgen Biotech experience gives all the materials for free, renting the equipment and all the reagents and everything. And they also do the curriculum and the training. And then NEB does free reagents too. If you send them your protocol, they'll send you free restriction enzymes and tech polymerase and things. There's an equipment depot. There's one in the Bay Area. They have laminar flow hoods and autoclaves now, and for free. It's for plant tissue culture, but they have a lot of stuff. So if, for equipment, the equipment depot has a lot. And then in terms of time, I was looking at for high schools, Carolina Bio has a full year-long course in a box. I think it was like $6,500. But that's so simple to just place one order and get all your reagents for the whole year, and then they give you vouchers so you can order it more timely. But thinking about high schools, how streamlined that process would be. So that's one thing I found I wanted to share. And so we've leveraged our external partnerships. We've worked with workforce investment boards. We're recently working with a family center, so El Nido Family Centers. They work with teen parents and also very young parents. They really want their the people that they serve to get vocational training and get into high-paying careers fast. So they're really interested in what we're doing. We've worked with other nonprofit organizations and the industry, and then, of course, our high school partnerships. At our campus, we've worked with the people who do educational partnerships with the high schools. We have a STEM center that we give presentations to in the STEM club. We've talked to our offices on campus for special populations, veterans and things, and talked to our counselors. I want to thank my dean, who's been very supportive, Gerson Valle, and then also my former dean, who's now at a different college, but he's been really helpful. So if anyone has any questions, if you want to follow up and continue the conversation, feel free to email me. Thank you.
Katharine, there were a few questions in the chat that were really more for your area, like, for example, the textbook and a couple other things. We had some articulation questions as well.
Okay, let me check that.
Thank you, Katharine. We now have time for a Q&A for both of our ATE project talks today, both Dr. Porter or Dr. Marsh. And I did, as Dean Valle pointed out, there are some comments in the chat from Linnea and Yingsu.
Okay. Yeah, the textbook was, it was, who is it, Jack? It's online. He adapted the OpenStax one to, and to build in Biotech. I have to look that up, what the last name of the author was.
O'Grady.
Yeah, thank you. O'Grady, yeah. Oh, Jim Hewlett is not named on the grant. He's paid as a consultant.
Ah, okay, got it.
High tech, I don't know. I need to check. Maybe.
What if we invited you?
Oh. Yeah, where is it? When is it? I'll have to look that up.
It's in Austin, end of July.
Oh. Maybe. Yeah.
Yeah. Check it out. We're going to have a biotech pre-conference and then throughout the high tech. I think you might benefit. You benefit our group. But I know, Katharine, you're a busy woman.
Yeah. Okay, I'll check that out. Thank you. The buses were paid by the high school. They did all that. I don't know. Yeah, we didn't pay for that.
I think you should write another grant. You have a lot of things that you still want to do. Always thinking grants is one after another.
Well, we just started another one.
Yeah, I was about to say she did write another grant, and it says just started. So.
Okay, all right. Well, then, see, I won't then put a third one on you. But good, excellent. No, I think it'd be great if you come to high tech and share some of your expertise.
Okay.
And Rebecca had a question in the chat. Are you willing to share the case studies for the quality class?
Yeah, I'll have to think about, I think so. I'll have to think about how to make sure students don't get access to that, but there probably is ways to do that. Yeah, follow up with me over email.
You can do that through Innovate Bio too, because we're set up to do that. So Sandy can help you with that.
Okay.
We have curriculum that we share, and then faculty can access it, but not students.
Oh, that's good. Because that'll help with the dissemination piece if I share it through Innovate Bio.
Yeah, we'll gladly do that for you.
Okay.
And Karen has a question for Sandy. Is there a plan for sustaining the hackathons going forward, or guidelines for others to try to replicate the model?
Yeah. As far as guidelines, we did publish a paper in JATE on the hackathons, and there's some guidebooks, but I have been thinking about writing a paper with all the team leaders about all the things that we've learned, because we've learned a lot over the 5 hackathons. So we're thinking about more work to write things up. Actually, believe it or not, people have replicated it already somewhat to some extent. There's a group at Florida Atlantic University they contacted me, and they started their first biotech hackathon in 2023. They get all their faculty to participate, and they've been collaborating with the local community colleges like Palm Beach State College has participated with them. And they've been their teams are developing like ideas for biotech products, kind of like the community college challenge a little bit, innovation challenge, except in a shorter period of time. As far as going forward, we're thinking about it. We turned Biotech Careers into a nonprofit, and one of our industry scientists mentioned that his company might be willing to sponsor hackathons. So we might do some of this through Biotech Careers. But right now, it's something we're thinking about. Thanks, Karen.
And I just want to say we really would love it if people would participate in the Innovate Bio Hackathon, because especially on things like the next center. Remember, this is our last center. Now, who's going to do the next center? And what's it going to look like? I guess Ying Su has her hand up. I don't think it's my role to call on her, but
Dion already called. Dion, you're calling on me, right, Dion? So for Katharine and your dean, Compton, what is that school called? Compton Tech Innovation. They're very interested in working with you guys. They are starting their charter school. So the first year is 9th grade, and then 10th grade, 11th grade, 12th grade. But part of their curriculum is introducing students to all the different STEM programs that are out there, and one of them being a biotech unit. So they're already starting this with their students, and what they want to do then is to build out a pathway for biotechnology at their school and in working with you guys. So I don't know if you have contact with them. I know the principal, she's awesome. And so it's something to think about with that charter school, because they're very focused on STEM, and biotech is one of them.
Hi, Ying. We do have talks with them. We do our dual enrollment coordination approximately a year out. So now that they're starting to get ready with biotech, most likely we'll start that next year.
Okay, great. Yeah, and we did provide them with some equipment. We had equipment that was lent out to a school down south, and so we were able to provide it for them for a much lower cost because it was used. And so they have micropipettes, and I think they have a transformation kit, which is a water bath and incubator.
Sounds good.
Yeah, so if there's more equipment that they need, we can also help them with getting some as well. Because mini ones right now are $1,000 off for a set of 10. And I told them that too. So the principal knows about that, and you have to just buy it before June. And it's a really good deal for 10 electrophoresis for under 2,500, total.
Yeah, we are in talks with them. Currently, right now, we are starting a biotechnology program pathway with Fireball High School, which is in a neighboring district as well.
Great.
yeah.
Thanks.
And yes, that's how we get our dual credit supporting, because all our high schools run their own courses. We don't have any at the campus, and we use our students in the 2-year program during internship to help with the high school courses. It's really good because they have a lot of buy-in. And then if the high school students show up in our courses eventually to get the 2-year degree, then they like giving back by doing their internship at the high school they came from.
That's really cool.
No one wants to leave our program.
Are there any additional questions or comments for either of our Project Talk speakers?
No, I was inviting people to high tech, so I hope you don't mind, Cassandra. We still have spots.
Kara, I'm sorry, were you about to?
Yeah, I'll jump in with a couple of questions. We're in an area that's just starting to get their biotech programs going. And so I would appreciate any resources. One we're working on right now is just more forming like the needs analysis or jobs data analysis. And then of course, we're going to be starting some pilot programs. And so that open source software or any tools would be really helpful. So, thank you.
Innovate Bio can help. We can send you a lot of curriculum for free. We can send you entire programs if you're interested.
Yeah, that'd be great. So Innovate Bio has that resource?
Yep.
Okay.
Yep. You can look us up, innovatebio.org. That's our job, is to help. And where are you?
So I'm in South Dakota, and
Oh,
the
wow.
area of biomanufacturing.
We want you to come to high tech.
Yes.
Okay.
I did take a look at that. And
Please.
so, yeah, and the area we're focused on is more bioprocessing. And so more the industrial ag focus of biomanufacturing, but I know there's a lot of crossover.
We have a lot of curriculum for biomanufacturing. You should not have to create any of it yourself.
Yeah,
And we have the skill standards too.
For that, you want to go to biomanufacturing.org.
Okay.
Mm-hmm. Although, isn't the company that's in South Dakota, don't they manufacture a lot of plasmids? I mean, it's going to be a little bit different.
There's a few different companies. The one I'm working with right now is Dakota Bioworks.
Mm.
So,
do you want to schedule a meeting with Innovate Bio and we can talk?
And the Nebraska people too, and Iowa.
So I'll put in my email and let's schedule a talk. And Cassandra and Dion can help with this, but we should schedule a talk about how we can help you and what we can send you. Okay? You see my email?
Great. And then that website, Todd, was like biomanufacturing.org.
I'm putting it in the, okay.
Yeah, Sandy put it in.
But we'd appreciate, they're a hub for us. Why don't you contact the center too? Because we can even put you into more resources than just biomanufacturing too.
You bet. Thank you.
And we have a couple of more minutes left. I want to direct you to the chat. Sandy shared the high tech link in the chat, and also there is a link for the Innovate Bio newsletter sign up. Please become a member of our community. And lastly, there is an evaluation survey link. We ask that you complete that link, and that helps us with scheduling future events. So again, please join me in thanks to both of our presenters today, and thank you all for joining and contributing to our discussion. We wish you all a fabulous weekend, and I'm sorry. Oh, Todd, that's a kudos hand. Good.
Yeah. And Kara, I think, wouldn't Kara be our first program